Okay, now we will study the next one, the in vitro method. In vitro method is nothing but that is outside or in the glass or glass tube or test tube, or we can call as a uh, petri dish, like that, something. The cells we are separating, what are the things here we are using in vitro methods, specifically cells, enzymes, or any tissues we are separating from the body, that is stored in the lab in the controlled environment. In that controlled environment, we are giving our new trial drug. Whether the drug is acting or not, we are checking in the glass slide or petri dish. In that we are analyzing. This is technique is in vitro method. Here lab based study, this is the preclinical lab based study here, performed outside the living organisms. It means that we are separating the here cells we are separating, or else tissues or enzymes we are separating and using in the controlled environment in the lab, specific environment. We are analyzing whether the drug is acting or not. Example we can take, that is the ashwagandha withenia somnifera root extract. That root extract we are separating here, tested on human cancerous cells to assess the cytotoxic activity. What it means? The simple meaning is that the in vitro methods, the cancerous cells we are separating in the test tube or petri dish, that we are keeping in the controlled environment. After that, the ashwagandha withenine is phytoconstant. We are separating this chemical constituent, we are separating and adding in the this cancerous cells. Whether the drug is effective or not, we are checking in the in vitro. If the drug is kills the cells, or the drug after the using of this withenine, the drug, the cells, enzymes or tissues, there dies, it nothing but the this is one of the cytotoxic drugs. It means that you can use the drug in the cancerous cells. Like that we are analyzing the in the lab, in the petri dish or in the test tubes. We are checking whether the if you are using this drug, whether it is safe for the cells or tissue or not, we are checking here in this methods. Okay, this is the example. Next is the applications. Here application is the cell viability assays. What is cell viability assay? Just now I told whether the your new trial drug is safe for the cell or not, that we are analyzing by using in vitro method. This is the cell viability. Next one, enzyme inhibition study. You know, that is the whenever we are separating the enzymes, the most of the due to the enzyme that so many diseases are produced. Whenever the new your trial drugs, the if you are giving here, the checking the enzyme inhibition, the drug is inhibit the enzyme or not, that we are studying by using in this method, in vitro method. Next one is the receptor binding assays. This is a very important method for analyzing the in the research, the receptors binding. It means that you know what happened whenever you are giving the your trial drug, your main important the trial drugs, whether it is binding with the this receptor or not. If binding it, then only it will work. That also helpful for the assess or analyzing in the in vitro methods. The your drug is whether binding with the particular receptor or not, that we are analyzing in this method. This is the in vitro methods. Now advantages. What is the here advantages? High throughput it is there. It means that the so many things we can do at a time. This is a very high thoughtful process you can do with the in vitro method. It means the fast you can at a time you can do so many high thoughtful process. And next, reduce animal use. No, that is the very problematic, that using animals or human beings. This is the one of the safest, the reduce the animal use here because of this in vitro methods. Now the limitation. What are the limitations? Why some people they are not believing here? It does not replicate full body interactions. It means that only you are separating the cells, tissues or enzymes only. We cannot completely tell that whether drug is acting or not, because only simply we are testing on the only simple cells. That's why it is does not replicate full body interaction. This is one of the limitation in the in vitro method.